Assalamu alaikum students, good afternoon. In the last lecture we have completed the living world and we enter into the biological classification. वहाँ मैंने biological classification में आपको जो है introductory part बताया और हम फिर जो है kingdom monera में आ गए, ठीक है ना? So we are continue with the kingdom monera. Continue with kingdom monera. Okay, so in the kingdom monera, the main representative of the kingdom monera is the organism which represents this kingdom. It is the bacteria. Okay, so bacteria is the sole organism for the kingdom monera. So whatever the characteristic features are present in the bacteria, those all characteristic features we can consider they are present in the kingdom monera. Okay, so let me take you through first the structure of the bacterial cell. Okay, and all features which are present in the bacterial cell, they all are representing the kingdom monera itself. Fine. Okay, so let's talk about the structure of typical bacterial cell. Okay, so if I draw the structure of the bacterial cell has the outermost covering, envelope system by which the bacteria is covered from the outer side. Okay, and this envelope system has different coverings of different layers that I will label also. Fine. Okay, so this is the outermost covering, outermost boundary of the bacterial cell. Okay, the outermost layer of bacterial cell, this is called glycocalyx layer. It is known as glycocalyx layer. Fine. Now after the glycocalyx layer, inside the glycocalyx layer there is another layer present in the bacteria. This is called cell wall. Fine. Inside the cell wall there is a presence of another boundary, another membrane. It is known as plasma membrane or cell membrane. Fine. So by these three layers or three covering, the whole bacteria is covered from externally. Okay, so that is the envelope system of the bacteria. Externally, if we see what are the structures present on the bacterial cell, externally there are certain structures present externally on the body of the bacteria. Hair-like structures are present at different places. Some hairs are smaller and some are larger in the size. Okay, like this. Some are larger in size, some are smaller in size, like that. Okay, these hair-like structures are outgrowths which are present on their body. They are known as either pili or fimbriae. Structurally they are similar, but functionally they are little bit different. They do the different function which we'll discuss later on. First let me show you the structure of the bacterial cell. Okay, so these are known as pili and fimbriae. Externally on their body they have also the presence of some locomotory organs which are present in them, those locomotory organs get arised from the centriolar like bodies inside the cell membrane, which led the formation of structure, a hook like structure then from outside of their body, and a long elongated tail like structure is being formed like this. Okay, so from where this whole tail like structure get developed from the two granular bodies, like these two granular bodies, they are known as basal bodies. They are known as basal body. So basal bodies are actually, they are the two centriolar bodies. They are the two centriolar bodies. From those centriolar bodies, the fiber-like structures get arising from them. The fibers get arising, and those fiber let the shape of plate of like structures, plate-like structure, which are present within the cell wall and cell membrane. They are known as basal plates. Basal plates. Fine. Then there is a hook-like structure being formed outside of their body. Like this is a hook part, that is called simply a hook. And from the hook, long elongated tail-like structure, it is called filament. It is known as filament. Okay. So altogether, by compiling of all these components, basal bodies, basal plates, all together they constitute a structure. That structure is known as flagella or flagellum. So this is known as the flagella. Okay, so externally on the bacterial cell, the bacteria may have presence of this flagella on an average 1 to 8 in number. On the bacterial body, bacteria could have presence of 1 to 8 number of flagella, but not more than 8 number. Okay, so similarly there could be the same type of flagella from the another side of the body of the bacteria, like this. Okay, so this is the again flagella. So externally the bacterial cell has presence of the envelope system, and it has the presence of the femorei, pili and flagella, which helps in the locomotion movement of the bacteria. Okay, now internally, if you see inside of the bacterial cell, this region of the bacteria, it is the cytoplasmic region. This is the main cytoplasmic region, which is the main arena for the metabolic activities. In this cytoplasmic region, bacteria, they have presence of genetic material which contains the DNA and RNA. Okay, this genetic material in the bacteria, it is completely naked. It doesn't have any envelope system. It is completely naked in them, and this genetic material is coagulated in the central location. In the center of the cell, it gets coagulated, and this region which contains the DNA genome of the bacterial cell, it is known as nucleoid. It has the other names also. It is also called genophore. It is also known as incipient nucleus. It has different names. Okay, genophore, incipient nucleus, nucleoid. So this nucleus, it is actually a naked genetic material which is not well developed. This doesn't contain nuclear envelope. Okay, it doesn't have nuclear envelope. It doesn't have a nucleolus also. Okay, so because of that, it is not well developed nucleus. So that underdeveloped nucleus, it is known as nucleoid, genophore, or incipient nucleus. Remember these terminologies. Okay, now in the bacterial cell, at certain places this plasma membrane, it shows invaginations at different location. It goes enfolded inside. It goes enfolded. Like for example, here the plasma membrane goes enfold like this. For example, there is I have highlighted this small portion here. Okay, this small portion of the plasma membrane. This goes enfolded inside, highly folded structure. Then it led a structure here, over here. That structure is connected with this nucleoid. It is connected with nucleoid. Highly enfolded structure of plasma membrane in the center of the cell, which is connected with the nucleoid of this region, nucleoid of the bacteria. It is known as mesosome. It is known as mesosome. The whole enfolding occurs at the center location, and enfoldings occurs at the peripheral locations also, like this. At peripheral locations, it also goes enfolded inside. Let some structures are being formed by this plasma membrane enfolding. Those structures are known as mesosomes. So if it is formed in the central position, so it is known as central mesosome. Central mesosome. Fine. And if the same mesosome is formed at peripheral location like this, then it is called peripheral mesosome. It is known as peripheral mesosome. Fine. Or peripheral mesosomes, they are also known as chondroid. They are also called chondroids. Remember these terminologies. Now, structurally, peripheral is different, central is different, but functionally also they are different. This is doing some function which is different from the peripheral mesosome, that I will explain you later. Okay. So, structurally, this is the modification of the plasma membrane inside. Now, inside this whole cytoplasm, in the bacterial cell, as you know that the bacterial cell is a prokaryotic cell. So prokaryotes, they do not contain any membrane bound cell organelles. The membrane bound cell organelles are completely absent. Remember this, the membrane bounded cell organelles are absent. Okay, except they have a presence of one organelle only, which is membraneless. That doesn't have any membrane covering. So that organelle is present in the prokaryotes, which is known as ribosome. It is called ribosome, especially 70S type. 70S type of the ribosome. Okay, so there are two types of ribosomes, 70S type and 80S type. Okay, 70S type ribosome can be found only in the prokaryotic cells like bacterial cell, and 80S ribosome is only found in the eukaryotic cells. Remember this. So this prokaryotic ribosome 70S type is present in the whole cytoplasmic region. Some of the ribosomes get associated with the plasma membrane nearby peripheral mesosomes, like this. Nearby the peripheral mesosomes, those ribosomes which get associated with the plasma membrane nearby the peripheral mesosomes, these functions as the respiratory enzymes. They function as respiratory enzymes. They function as respiratory enzymes, which helps the respiration of the bacteria. So their main role is to help the respiration, cellular respiration of the bacteria. Okay. Apart from that, apart from that, the bacteria does not contain any cellular organelle inside, okay, like mitochondria, chloroplast, Golgi body, ER, these are all absent in the bacterial prokaryotic cell except ribosome. Apart from that, the bacterial cell, the bacteria stores the food material. So the food is going to be stored inside the cytoplasmic region in different bodies, like some food is going to be stored in different bodies, like those bodies are called glycogen granules. Glycogen granules. So like the food is going to be stored in the form of glycogen, so they are stored in the form of granules, so they are known as glycogen granule. Similarly, the food is going to be stored in the form of phosphate granules. In the form of phosphate granules, they are called volutin granules. They are known as volutin granules. Volutin granules are PO4 like phosphate. They are the phosphate granules. Okay. Similarly, food is going to be stored in the form of fat droplets. Fat droplets or fat granules. Some of the fat is also being stored in the form of the food. So different food is stored in the different bodies. All these food storage bodies together, like fat droplet, volutin granule, glycogen granule, they all are known as inclusion bodies. इनको बोलते हैं inclusion bodies. So inclusion bodies are those bodies which are non-living in the nature. So all these food storage bodies are in the non-living nature, so they are called inclusion bodies. Fine, remember. Sometimes in the bacterial cell, in the bacteria during the process of protein synthesis, when the bacteria has to go for the protein synthesis, when the bacteria has to prepare the proteins, at the time those ribosomes, and there is a RNA molecule in the cytoplasmic region. The RNA molecule is required for protein synthesis. On a single RNA molecule, usually one ribosome is enough to decode the genetic information of the mRNA to let the formation of the protein synthesis. But sometimes on a single mRNA molecule, more than one ribosome is attached on the same mRNA molecule to decode this mRNA and let the formation of different polypeptide chains, which led the formation of the different proteins. Okay, so that structure where more than one ribosome is attached to the mRNA molecule at certain places in the cytoplasm, this structure, it is known as polysome. It is known as polysome. Remember, so that is called polysome. Okay, so these are also some structures formed within the bacterial cell. Apart from this structure, there are some bacteria, not all bacteria, in certain bacteria, apart from its own genomic DNA, this main DNA, central DNA, which is the cell's own DNA, apart from that, certain bacteria, they may have presence of some double-stranded circular DNA. Double-stranded circular DNA, and this double-stranded circular DNA also present in certain bacteria. It is called plasmid DNA. It is known as plasmid DNA. Okay, so this plasmid DNA can be found only in certain bacteria but not in all the bacteria. Remember this. Okay? So apart from that, inside the cell may have the presence of more than different types of enzymes which are very helpful to accomplish the metabolic reactions. ठीक है ना? So there are various enzymes. also present, which helps in the metabolic reactions of the bacterial cell. Fine. So this is the typical bacterial cell. What are the important features of the bacterial cell present in the bacteria? That whole thing represents the kingdom Monera itself. Okay, so that is the main important things to remember about the bacterial cell as well as the kingdom Monera. Okay, so I will give you the explanation of different all these coverings, their compositions also and their function also, that we'll discuss in the next class. Okay, so till then just be thoroughly about this topic or the structure of the bacterial cell, know about it. Till then I will give you the next lecture based on the explanation of the components of the different components of the different layers of the bacterial cell and inside the cytoplasm also. Okay, thank you so much.